So today we are going to see the topic antigen-antibody interaction. So we'll see about the whole antigen and what are the antibody and what are the types. So coming to the antigen. So antigen, it is also called as pathogen or foreign substances. It is a molecule that initiates the production of antibodies and it causes an immune response. So when the antigen attacks our body immediately involved in the production of antibodies through the antigen. So antigens are the foreign pathogens that invade the body and it is having the capability to give rise to the immune response to our body, and these are the large molecules of proteins which is present on the surface of the pathogen in any of the bacteria, fungi, virus and other foreign particles. So when these harmful agents that enters in our body, immediately it induces an immune response in the body for the production of antibodies. So coming to the properties. So the antigen should be a foreign substance to induce an immune response, and the antigen which is having the molecular mass of 14,000 to 6 lakh Dalton. So Da indicates the Dalton in which Dalton is expressed for protein molecules. So antigen is also a type of protein molecule. They are mainly proteins and polysaccharides. They are chemically complex. They are more immunogenic they will be. And antigens are species specific and the age influences the immunogenicity. So coming to the age, very young and very old people exhibit very low immunogenicity because there are too little very young and there are very too old, and old people and young people does not exhibit that much of immunity power when compared to other people. So coming to the antibody, so it is also called as immunoglobulin and it is represented by the symbol Ig, and it is produced mainly by the plasma cells or B cells or B lymphocytes. So antibody is mainly produced by the B cells. So it is coming to the shape, it is the Y-shaped structure made up of 4 polypeptide subunits, and each subunit has 2 identical light and heavy chains. So there are 2 antigen binding domains that forms the arms for the Y shape, and that site is called as a fragment antigen binding site, and so the C terminus of the heavy chains forms the fragment crystallization and which helps in the interaction with the effector cells. And these all 4 polypeptide subunits are held together by disulfide bond. So disulfide in the sense S bond S and no non-covalent bonds. And the heavy chains of the antibodies contains the variable regions and 3 constant regions. And each antibody has 2 identical antigen binding site and they differ in the antibodies. So mainly it is a Y-shaped structure and it has 2 subunits and 2 identical light and heavy chains, and it is having the fragment antigen binding site and it is attached by the disulfide bond, and it is having the variable region and constant region, and each antibody has 2 identical antigen binding sites. So this is the structure. You may able to find out the antigen binding site in both the arms, and this is the light chain, whereas H blue color H is the heavy chain and L is the light chain, and both the heavy chains and light chains consist of variable region is denoted by V and constant region is denoted by C, and these variable region and constant regions together forming the fragment antibody binding site. And you may see the disulfide bond which acts as the bond for connecting the 2 arms, where C is the constant domain, V is the variable domain, and H and L are the heavy chains and light chains, wherein you may find out the antigen binding site in the top of the 2 arms, where the outside antigen will bind to that receptor. So it is also called as antigen binding receptor.